him. So Joseph Amsili is an extension associate at Cornell, where he serves to coordinate extension and research activities associated with the Cornell Soil Health Program and New York Soil Health. Several current activities he's involved in include coordinating the New York Soil Health Specialist Training, which is a great program. I participated in the first round of it, the New York Soil Health Working Group, and statewide soil health characterization project. He completed his master's at Penn State, where he was a part of a research group studying the multiple benefits of cover crop monocultures and mixtures for farmers. His master's research focused on comparing cover crop root traits and on tracing cover crop rhizodeposition, which are the organic carbons released from living plant roots, into the stable fractions of soils. Joseph is extremely passionate about soil health, and his excitement is rooted in his passion for promoting sustainable soil management strategies. So thank you, Joseph, very much for speaking today, and I'm going to turn it over to you.
All right, Debbie, can you see my presentation there? Cool. Well, all right, everyone, it's great to be virtually on Long Island. It's the closest I've made it to Long Island, even though I've been telling Debbie and Anne Marie I would try to make it down there for a long time. I'm excited to be giving this presentation today on digging deeper. It was great following Debbie because Debbie showed a lot of above ground biomass pictures and some field stuff. And one of my passions has been to try to uncover some hidden relationships between cover crop roots and their functions, because a lot of what we know about cover crops and their benefits is from species identity or chopping the above ground portions off. And as anyone who's known that's done root work, it's really hard. So the purpose of this presentation is really to unlock the black box of cover crop root traits. So maybe this isn't a necessary part, but as you can imagine, we face a triple threat to global food security. Seen from outer space, humans have been a soil degrading species as our legacy on Earth. What we don't see in this picture is CO2 leaving the soil. So if we don't, important concept is if we don't add the same amount of organic matter inputs to the soil, we will slowly degrade our soil organic matter in the soil. And some recent estimates said that about 50% of soil organic matter in North America has been lost since we started agriculture in North America. We also have serious issues in terms of ag pollution, in terms of greenhouse gases that are released from agriculture and nutrient pollution. And we are faced with extreme weather events, including heat waves, droughts, like the one we're experiencing this summer, intense precipitation events that are predicted to increase in the future. And this farmer to the right, this is in 2016, a farmer in Western New York standing in his drought-stricken corn. So this was a 100-year drought in that year. So we're gonna be needing these triple threats really require sustainable soil management strategies that can address all these issues together. So why plant cover crops? A really great resource for understanding changing practices in the United States is this annual SARE CTIC cover crop survey. And this was a survey to about 2,000 farmers in 2016. And these were the reasons they listed. So their motivation for using cover crops, as many of your motivations for using cover crops include weed control, increasing cash crop yield, having diverse rooting systems, nitrogen supply, nitrogen scavenging, reducing soil erosion is always a really big one, reducing soil compaction, increasing soil organic matter, and this catch-all of increasing overall soil health. So because we're going to be talking about how cover crops influence soil health, let's define it briefly. You probably are all pretty familiar with this definition by now, but it's really about the capacity of soil to function. So when as a living ecosystem that sustains plants, animals. So some of those functions we really care about. The basic one, crop or pasture production, our plant productivity and plant health, you could call it. Those services and functions relating to water retention, transmission, and purification through the soil. The function that soil provides a habitat and a support system for life, which in turn supports plant health. And some people might be really interested in carbon storage. We're hearing a lot about carbon farming these days. These are the exact same two inherent soil types in New Hampshire, but you can see the range that these soils have been taken to on maybe a conventional grain operation and maybe a smaller scale veg operation. And understanding the soil processes that underpins healthy soil functioning is really key. So soil health, to have an optimally functioning soil, we really need all of these physical realm, biological and chemical, to all be properly functioning. So some of the processes that are important to consider: first, the physical, and a really good analogy for thinking about the physical structure of the soil. Those soil particles are the skeletal matrix, similar to the wood beams in a house, and the rooms in a house are like pore spaces. So we all know that we desire bigger rooms, and if we have bigger rooms versus a collapsed house, there will be more room for activity, more room for proliferation of organisms and living roots. We need a soil that's going to be stable and resistant to erosion, a concept we like to call aggregate stability, or many people have seen the slaking demonstration. And the inverse to stable aggregates is a soil that slakes apart, and now those particles are free to move with water in erosion. Soils are really unique in that the capillarity allows them to both store water and water, to hold water against gravity and allow water to move through. And we need water to move through so that air can also occupy those pore spaces. Although plants are photosynthesized, the roots are aerobic like humans. So in some of the biological processes, that whole living system underground is like us. They need to be fed to function and do all those things that we need them to do. And one of the big ones is decomposition of organic matter. A really important example is that when you send your soil in for test, a lot of people don't understand that there's no good test for soil nitrogen. And the reason for that is that 95% of nitrogen is bound up in soil organic matter. So for that nitrogen to be released inorganically, microbes need to process the organic matter and release it. So although you can do a PSNT or inorganic nitrate sample, those levels are so ephemeral that those aren't even included in a routine nutrient test. Another one that's really important is this maintenance of soil structure. We know that fine roots, arbuscular mycorrhizal fungi, are really important for building soil aggregates and helping maintain them. And a really important newer area of research, maybe in the last 30 years, has been our growing understanding of a diverse microbiome in the soil, and its role in suppressing disease in the soil. And we've known a lot about these. Everyone's probably seen the effect of pH on nutrient availability, so hopefully we can skip that. And so this presentation is really about cover crops, and they are very powerful tools for influencing physical processes in the physical properties in the soil, and biological, and also important nutrient aspects of important nutrient benefits. And this is an important segue until we get into the main part of the presentation, is that cover crop species selection, there's so many species to choose from. And unlike agricultural inputs, fertilizer that has one purpose, these cover crops are all multifunctional. Each one has benefits and drawbacks. It's much more knowledge intensive, includes thinking about narrow windows of time for field operations, like Debbie was talking about. Triticale is not good for a summer window, and vice versa. Buckwheat shouldn't be really planted as a winter annual cover crop because it really shines in that summer window. So for cover crops, they're biological tools, so they need to have contingency plans, adaptive management, and all that. And one really good way to think about cover crop diversity and when you're designing mixtures, rather than thinking about adding as many species you can to a mix, thinking about groups of species that are similar, and plant families are a useful group. So this is a framework that we used in my Penn State research group that I was a part of. Here you can see on this, we'll call this the x-axis, we have legumes, brassicas, and grasses. And evolutionarily, these have evolved to have very different traits and benefits that they could provide the soil. And there's some legumes that are winter hardy, red clover, canola, cereal rye, although depending on where you're at. And then there are winter killed cover crops. So this might be an important framework for designing mixtures. Maybe you would want to design a mixture that had some oats, which will have great fall growth, with cereal rye, which will have slower fall growth, but greater spring growth when oats is winter killed. And I'm not going to talk too much about this, but just want to bring you this to your attention, this resource from USDA ARS. It's our version of a periodic table for cover crops, has a lot of great information of cool season cover crops, warm season cover crops, grasses, legumes, and other dicot species. So that's a useful one. And now to the major component of this presentation, which is how do we assess which cover crops are best for different goals? And as we discussed, and Debbie and I both mentioned, a lot of what we know about cover crops are from above ground measurements, but what about roots and their traits? And does that information of cover crop roots bring to light new information that can help us select cover crops that will accomplish the functions or benefits that we most care about? So why should we care about roots? Because they're pretty. Now, not just that. One really cool area of research in soil science that got me really inspired to study this for my master's is this really cool area of research where they've traced both above ground and below ground parts of plants in the soil. And paper after paper has shown, that's done that, has shown that the carbon derived from roots, that root carbon or exudates from the roots, is preferentially persists in the soil compared to that shoot carbon. So although there could be more shoot carbon added to the soil than roots, these scientists were seeing that root carbon was sticking around for more years than the shoot carbon. So that's a pretty cool piece of research that really highlights the benefits of growing growing roots into the soil for increasing soil organic matter. We hear a lot about this. Roots are carbon pumps. We usually hear about figures on the higher end, but basically, and many people call this exudation, although it's other things as well. But basically, plants are pumping maybe from anywhere from 4 to 30%, let's call it 10%, of the total amount of their photosynthetic energy, and pumping it into the soil as carbohydrates or sloughing off roots or decomposing roots. That's another reason why roots are important. Roots associate with bacterial nitrogen fixers and arbuscular mycorrhizal fungi. So there are important relationships there. We know, for example, that most brassicas are non-hosts of arbuscular mycorrhizal fungi, whereas grasses and legumes are hosts. We know that roots, fine roots, and arbuscular mycorrhizal fungi are really important for building and maintaining aggregates, which are important for that surface soil structure and the ability of water to either go through the soil or erode. And another important function that we think about is whether certain roots are better at breaking up through compacted soil. So we're going to talk about some of these different angles today. And I'm going to go quickly through this. This isn't as important, but some of the traits that are important in thinking about how the cover crop relates to the function. You might think of the total root biomass. One that you might not have heard of is this concept of root length density, which is basically the total amount of root length in a given volume. And in some of Debbie's images, you could see that some of those species probably have higher root length density than others. Probably buckwheat has higher than some of those legume species. Root diameter, we know that eudicots, as they grow, there's two types of plant groups, monocots and eudicots are characterized by having that widening of the root diameter, like a taproot. Root distribution, how they grow horizontally and vertically. The carbon-nitrogen ratio is critically important for dictating whether the organic matter residue will lead to nutrient release or nutrient tie-up. As I mentioned, mycorrhizal colonization, BNF, are bacterial nitrogen fixation. Are the plants actually fixing nitrogen? They might have the nitrogen fixer, they might not, or there might be so much nitrogen available in the soil that it doesn't make sense to invest in that relationship. And the word that I like for explaining exudation is this term rhizodeposition, because it's really a catch-all definition that describes the amount of carbon coming from living plant roots, whether it's from exudates, dying roots, you name it. So the first area we're going to talk about is how cover crops add organic matter between cash crops. And this organic matter addition stimulates biology, can maybe suppress disease, and decomposition can either increase nutrient availability or maybe retain nutrients in microbial biomass. So this brings me to where I did my master's research at Penn State. This is an aerial photograph of where I spent a lot of time in this big 20-acre organic corn, wheat, soybean rotation that was looking at cover crops in detail after winter wheat and before corn. They had done an earlier iteration of the experiment where they had all these same treatments in between corn and soybeans. But as we know, after corn comes off so late that there really isn't a long enough window, even in central PA, to establish really diverse, robust cover crop mixtures. So the experiment, most of the work we were doing when I got there was pretty much after winter wheat comes off in July, and you can really have a really nice window to plant your cover crops mid-August and take them off in early May before corn. So in this experiment, we were comparing 6 different monocultures, 2 brassicas, 2 legumes, and 2 grasses. And one of those was winter hardy and one of those was not. And then we had some mixtures combining those species, because all over the country, there's a lot of hubbub about mixtures. But this experiment was really, where does the rubber meet the road in terms of the relationship between species diversity and the amount of benefits we get from cover crops. So these were some of the treatments I looked at. Because I was looking at roots, I didn't want to look at all 12 treatments or else I would have gone crazy. But I looked at a legume, crimson clover, which is the most widely planted legume in the country. a winter annual cover crop. And this is important to mention, these are all winter annual cover crops, unlike the summer annual cover crops Debbie was talking about. So I also looked at triticale, which is a mix of cereal rye and winter wheat, and canola, which is not so common in New York, maybe more common in Long Island, but is the plant that we extract a lot of oil for. It has lower acid compared to some of those other mustards. And then I looked at a 3-species mixture that contained these 3 species. So fast forward through all the work and the methods to the results. So here's the fall data for that. And what was really cool to see, a lot of the big differences that I saw came down to different surprising traits of triticale. Triticale in this study in an organic system, maybe where nitrogen is limiting, had an extremely high root to shoot ratio. So there's about an equal amount of roots below ground than there were shoots above ground. And that's, I think, pretty shocking. And there were pretty large differences between the total amount of root biomass and the surface roots. All the treatments had more surface roots than crimson clover. So crimson clover is great for nitrogen fixation, but in terms of adding root biomass to the soil, it came up shorter than these other species. And another really unique trait that I think is really useful for understanding the linkage between root biomass and different traits is that triticale produced about 2 to 3 times more root biomass than other cover crops at all the depth intervals I looked at in both fall and spring. So this might have really important implications for thinking about nitrogen scavenging in the between row space. You're adding more carbon into that between row space, competition between weeds compared to these other species. So those were all important traits that I looked at. So if we were going to rank those in thinking about high root biomass is the best, it would be probably triticale had the highest root biomass. Canola and the mixture were intermediate, but they really didn't have as much roots in the between row space than triticale. But we also really need to factor in the effect of all this root biomass on the next cash crop, because that's really where the rubber meets the road. And time again, people find that cereal rye or triticale, these cover crops that are really good at putting on a lot of biomass, because they have a higher carbon and nitrogen ratio, can lead to a yield penalty for the following cash crop, especially in organic systems where you have less added fertility. So you need to think also from another perspective of thinking about the carbon and nitrogen ratios of these materials. And now a high carbon nitrogen ratio is really a drawback, where a low carbon nitrogen ratio is desirable. So for a long time, farmers have known that making a biculture of a grass and a legume is a really effective way to combine some of the strengths of those 2 plant families and also limit the drawbacks of each of those plant families. So now I'm going to transition into cover crop roots and their relationship with arbuscular mycorrhizal fungi. This is a really stunning photo from Dan Mobius-Clune, who used to work with us in the Cornell Soil Health Group. And I believe these are carrot roots, and they're arbuscular mycorrhizal fungi. So if you don't know about this relationship so well, here's the short version. Basically, roots, not brassicas, but roots to access nutrients like phosphorus and zinc that are really immobile in the soil. This relationship, which is maybe 400 million years old, developed where basically the arbuscular mycorrhizal fungi act as this continuation of the root and give you much more surface area to extract those poorly diffuse nutrients. P and zinc are two really important of those. And in return, some of those photosynthate, those carbohydrates from the plant, are shuttled to those mycorrhizal fungi, and they have a nice relationship that way. So when we think about species effects on AMF colonization, you might have some idea. But this was a study also a collaborator of mine did at Penn State, Ebony Murrell, who now works at the Land Institute. She's a really great scientist. And they basically looked at the percent AMF colonization in maize in all these plots. So this was after all these plots were grown, corn was planted everywhere, and they went to each of those plots and looked at the corn roots and used the method to determine how much percent of the root was colonized by arbuscular mycorrhizal fungi. And similar to some past work that has shown that really grasses are the best hosts for arbuscular mycorrhizal fungi. And we know that brassicas, canola and radish, are non-hosts of AMF. So here, we're showing that the grasses were the best hosts for AMF in basically supporting those arbuscular mycorrhizal communities in between in this cover crop window. And then when the cash crop's planted, there's more of that inoculum to infect, or not infect, but associate with corn roots. And if we were going to categorize this, I would say that grasses, then legumes, improve AMF colonization compared to brassicas and fallow. And there's also some, well, I guess I'll go on to the next one. A really important constraint in agricultural systems is poor aggregation. We know that from the really intense impact of rain that has on the soil, can lead to situations, maybe you just have plowed a field, have fit a field to plant something that needs a really nice seed bed, like beans or something like that, and then you have a torrential rainfall like we had today in Ithaca. And then what happens is basically all those particles break apart, and a lot of the silt particles migrate into the soil and clog the soil. And you can get these really nasty crusts, which can actually really prevent seedling emergence in the spring. And we know through research on the effects of plants and roots and arbuscular mycorrhizal fungi that at a large scale, the two big actors are really fine roots, and mycorrhizal fungi. At smaller scales, polysaccharides and things like that become important. But at this large scale, roots and hyphae are really important. So here we have our image of cover crops stimulating soil biology. A big AMF are big players, and the roots and AMF work together to help enmesh and entangle and kind of glue these aggregates back together. And that improves pore structure, allows water to infiltrate into the soil where we can use it versus running off the surface. So here's another study that was done at University of Guelph in Ontario. And they basically looked at the effect of some different cover crop species on wet aggregate stability. And in this data, you can see that sorghum Sudan grass, which we saw produces a lot of above ground biomass, also produces a lot of roots. They produce these really cool anthocyanin colors that dye the roots reddish. And then also annual ryegrass is known to have a lot of surface roots and be really densely rooted near the surface soil. And both those led to a small increase, a measurable increase in aggregate stability compared to some other cover crops. And basically these grasses, which are known to be the best hosts with AMF, also had more root biomass. Those two traits were really driving this increase in aggregate stability, I think. So another really important constraint in agricultural systems is soil compaction, which can be surface compaction or it can be subsoil compaction via plow pan, which both limits root growth. There's certain points that it really is too energy intensive for roots to grow at that through that at that pressure through the soil. So it really causes them to invest a lot of energy that they shouldn't have to do in that. And infiltration of water, obviously. And you can see the differences of root proliferation in soil with more pore space, more macropore space versus root proliferation in a compacted clod here. You only see one root. And the big difference between here, you can think about all the nutrients that would be accessible here if the roots were able to grow there. Whereas in this picture, the roots are able to access the total soil volume and take advantage of all the nutrients and water in that soil. So this is a picture of a penetrometer, which is a useful tool to assess surface and subsurface compaction if you don't have a lot of rocks and if there's some adequate soil moisture. So if you did it now on Long Island, it would be way too dry and the soil would be like a brick, and this wouldn't be a useful tool now. And there's been some cool research on certain cover crops being better adept at growing through compacted soil than other cover crops. So that breaking up compaction can also improve pore structure and allow more water to infiltrate, but we also have this, that those pore spaces can allow other roots to grow in, and they can access more nutrients and water. So these are some images taken from Ray Weil, who is at University of Maryland, one of definitely a cover crop guru, who's been studying the effects of cover crops and them as tools in sustainable agricultural systems for a long time. So here's an image they took in 2002 of canola root growing through compacted soil. And lo and behold, next after the cover crops terminated, soybeans planted, they saw that the soybean, rather than try to move through the compacted soil, is really taking advantage of these preferential flow paths, preferential rooting channels that were created by the canola root. And that's really because it's not as well adapted to move through compacted soil. It really will always take the path of least resistance. And here's some other images of that, of these root channels made by canola and soybeans. Just followed it. And this is a very intensive way to study roots, but basically they install these tubes in the soil and drop a camera down at different depths and take pictures. It's really one of the only ways to study in situ root processes. So this is another really cool study that they did. They simulated different levels of compaction in a field setting and had different cover crop treatments in those different compaction levels. So in the high compaction zone, and this is cover crop root number. So this is basically, higher number is more roots, to make it simple. But the FR, which is forage radish, rapeseed, which are two brassicas, and cereal rye, which is a grass. In this most compacted situation, they were seeing that both forage radish and rapeseed were able to proliferate at have much higher root biomass going down the soil profile. On the surface, maybe cereal rye had much more, but as soon as you got 5 centimeters down or 10 centimeters down the soil, it was winning out. And similarly with moderate compaction, similar things, both forage radish and the rapeseed having more root biomass with depth and that moderate compaction. And then when we had no compaction, cereal rye was similar to rapeseed in the amount of roots that produced. Maybe more roots at the surface, which is a similar finding from my study, but interesting results. And then this is some really cool work that was done in England, where they actually took CAT scans. This was probably expensive, and similar to Ray Weil's study, they simulated different levels of compaction in these tubes. So the top part of the tube had bulk density that was about 1.2 grams per centimeter cubed, and then they increased that in the lower part of the profile. And they did some CAT scan images, and here are some of those 3D images. And you can see that these forage radish, that the taproots were able to grow through that compacted soil. Hairy vetch, which is a really important legume, winter annual legume, was not able to send many roots down. And this black oak was able to send a little bit down. So these, similar to our, maybe you could summarize this. I'm making generalizations. There's always the danger that there's a species exception, but here I would say brassicas are the best at growing through compacted soil. Another exception is sorghum Sudan grass, which is really good at breaking up through compacted soil, and then grasses. And legumes, I think, are the most lame for growing through compacted soil. So some key take-home messages. Certain species can, like triticale, some of these grass species, I would also suspect the same would be for cereal rye. I think it depends how much nitrogen is available in the soil, but in maybe an organic system, you can have very high root to shoot ratios below ground. So even if you're seeing a pretty measly 1,000 pounds of biomass per acre, there could be another 1,000 or 850 pounds per acre below the ground. And a really cool trait from my master's research was showing that the lateral roots in the between row space, there was maybe 2 to 3 times more than some of those other cover crop species. Grasses followed by legumes. Grasses are the best, followed by legumes for improving AMF colonization and really keeping those AMF inoculum in high numbers for the next cash crop. And we know brassicas are non-hosts and no cover crop. No cover crops and fallow periods and tillage are known to destroy the inoculum. We know annual ryegrass, sorghum Sudan grass, they were able to improve egg root stability due to their large root mass and probably a lot of root length in the soil. From some research that's been done, brassicas and sorghum Sudan grass appear to be the most effective at growing through compacted layers in the soil. We hear a lot about forage radish at being effective at this, sorghum Sudan grass as well, and those other brassica species are good as well. And it's really important, I think, knowledge of these root traits of these different species adds another angle to thinking about the multifunctionality and the benefits and disservices and drawbacks that each species brings. And for example, for AMF, if you're just growing a forage radish, then you're really not having species in there that are supporting that arbuscular mycorrhizal community. Maybe you want the brassicas to break up compaction. It really depends on what your goals are. But if your goals are to have a little bit of all services, then maybe mixtures make sense. So now we're going to transition into this part of the lecture called the cover crop root lab. And this is basically, we did the same thing that Debbie did. We grew some species, cover crop species, for about 2 months in PVC tubes, and then washed the roots. So this is about 2 months of growth in the greenhouse. And So yeah, some of these species are also summer annuals, winter annuals, but we'll start with the grasses first. So this is cereal rye, which is a really important winter annual grass species, cool season cover crop that would be planted, that can be planted the latest out of any of these cover crops. So it's usually the last option for establishing a cover crop. And here you can see really a lot of root biomass production, like similar to triticale, a lot of fine roots and really holding that soil together. Here's triticale, which is very closely related to cereal rye, cross between cereal rye and winter wheat, and also really impressive root biomass production and above ground biomass production, but in the species like this, you might expect a higher root to shoot ratio. This is annual ryegrass. This is known to be, produce a lot of really fine roots, and you can see you can see really how much fine roots there are. It's known to be really good at improving aggregate stability in the surface soil because there's so many fine roots. That's pretty impressive. Sorghum Sudan grass is a summer annual cover crop. And I talked about these anthocyanin pigments, really cool orange colors. And when Debbie had the picture, there was a really cool contrast between sorghum Sudan grass and pearl millet. Pearl millet didn't really have these really thick, wiry, adventitious roots that are probably one of the reasons that sorghum Sudan grass is so good at growing through compacted soil. You can think of these more wiry, greater diameter roots being really important for being able to grow through compacted soil. Whereas maybe the fine roots don't have that pressure and strength to be able to grow through something so abrasive and harder. So buckwheat, another summer annual cover crop. Great weed suppressor, like Debbie already mentioned, but really needs to be planted in those peak hot summer months to be effective weed suppressor. If it's planted out of that window, it won't be an effective weed suppressor. And these fine roots are probably the reason that buckwheat is known so well for being such a good phosphorus scavenger. Having as much root length per volume of soil really helps because basically the phosphorus only can diffuse so far in the soil. So having as much root length as possible with our arbuscular mycorrhizal fungi can increase the amount of surface area that you're able to access phosphorus at. So that's probably a good reason why buckwheat is such a good phosphorus scavenger. Buckwheat's another one, and it's really hard to wash these, or forage radish is another one. It's really hard to wash these roots because some of these brassica roots, I think, some of these fine roots are much finer than the roots you see in grasses or legumes. And one evolutionary hypothesis for why that is that because brassicas are non-hosts of AMF, they need to have another way to have really fine absorptive surface area to extract things like phosphorus and other things. Forage radish and brassicas are really effective nitrogen scavengers due to having all this fine roots. If there's a lot of nitrogen in the soil, one thing we saw in some of our Penn State work is if there's a lot of fertility in the soil, brassicas will always dominate. They'll outcompete other species, put up their big canopy, and shade other species from growing. So they're very competitive. Here's yellow mustard. Sorry, things are a little laggy. These pictures are so high res. So here's slightly smaller taproot, but similar to forage radish, has these really fine roots. And this is one maybe like the caliente that could also have those biofumigant properties. If you compare that to canola, basically those biofumigant properties have been bred out so that it's better, it's a more appropriate for extracting oil that's lower in acid for cooking. Actually, where canola, the name comes from, is Canada oil low acid, which is interesting. This is crimson clover. And definitely if we compared it to, did a non-scientific comparison, just looking at one replicate of this, much less root biomass than those grass species, but a very common winter annual cover crop. Has fast growth compared to red clover, which is maybe more of a biennial perennial nature and has a little bit slower growth habit. So yeah, crimson clover is a good one as well. Hairy vetch, in terms of producing root biomass and compared to crimson clover, I think is much more robust root biomass producer. And hairy vetch is a really nice cover crop, except for wheat farmers don't want to go anywhere near it because some of the seed basically won't germinate when you, some of the seed is hard seed, what they call hard seed, and it won't germinate immediately. And basically you're left with maybe the seed germinating later, and then it actually has the same size. So it's very hard to sort from wheat seed in the grain sorting operation. So hairy vetch, sorry. This is cowpea. And I should probably wrap up soon, so I'll start going through these quicker. Cowpea, summer annual legume. Sun hemp has these really bizarre, huge nodules. For some reason, the nodules aren't normal size. They're jumbo, tropical size. This is red clover. And you can see compared to some of these other roots that these are a little bit coarser, less thin than some of those brassica roots. And that's, I think, a generalization that's appropriate. This is alfalfa, perennial legume. And yellow sweet clover. So maybe I'll wrap up with this really cool story from University of Wisconsin-Madison. So yeah, this is a cool story. So maybe in the 50s, a farmer was feeding his cows yellow sweet clover hay, and basically some cows in Wisconsin were dying, and eventually a farmer was puzzled by this, took a pail, of blood to the University Wisconsin extension agent and said, why isn't this blood coagulating? This is really strange. So this actually led University of Wisconsin to realize that a compound from this decomposing, fresh sweet clover hay is fine to eat, but in the decomposition process, there was a compound that was released, which was stopping the coagulation of blood. So this led scientists to basically use that both in rat poison and as a blood thinner, and has led to a lot of royalties for Wisconsin-Madison, all because cows were dying because their blood wasn't coagulating. So that's, I'll skip this because I went over. And yeah, thank you so much for listening in. It's always hard to gauge the audience when you're virtual, so I appreciate you bearing with me. Here are some great resources. New York Soil Health Listserv is great for getting out information for events like this. Yeah, and thanks so much.
Thanks very much, Joseph, for the great presentation and some really excellent pictures. I find the pictures and just your research results very, very fascinating. So thank you for taking the time this evening to do that. It's certainly tough when you can't see people, and we'd all prefer to be in person, but glad you could virtually come to Long Island. So that's great.